Hello, everyone, and welcome to today's presentation. I'm excited to have you here as we explore the fascinating world of pharmaceutical drug extraction, upstream and downstream technologies. On this first slide, we see our main focus, transforming biological systems into life-saving medicines through the power of precision engineering and biotechnology. Over the course of this talk, we'll cover how these advanced processes work together to develop effective therapies. So, let's dive in and discover how science and innovation are shaping the future of medicine. Let's take a closer look at the biomanufacturing journey, which actually consists of two critical phases that work hand in hand. First, we have upstream processing. This is where we grow and nurture biological cells in carefully controlled bioreactor environments. These cells are essentially the factories that produce the drug substance, so creating the right conditions is key to ensuring they produce high-quality material. Next, we move to downstream processing. This is where the real refinement happens. We separate, purify, and formulate the drug into its final, usable form. This involves sophisticated separation techniques that ensure the product is both pure and safe for patients. Both of these phases are interconnected, functioning together in harmony. This synergy is what allows us to achieve consistent, high-quality pharmaceutical production at a commercial scale. It's all about balancing growth and refinement to bring effective medicines to those who need them. Let's take a closer look at upstream processing, which is essentially about cultivating our biological factory. The first step is cell line development, where we select and engineer the cells that will produce our drug. This involves optimizing these cells to express the desired molecules at high levels, ensuring efficiency from the start. Next, we move to media preparation. Here, we create nutrient-rich environments, think sugars, amino acids, and minerals, that fuel the growth of these cells. It's like providing the perfect diet to keep our biological factory running smoothly. Then we have bioreactor cultivation, where we control key parameters such as temperature, pH, oxygen levels, and agitation. Managing these factors carefully is crucial because it directly influences how well the cells grow and how much of the product they produce. Finally, we have fermentation scale-up. This is where we transition from small lab experiments to large-scale industrial production. Scaling up allows us to maximize capacity, ensuring we can meet the demands for our drug. All these steps are interconnected and vital for producing high-quality biopharmaceuticals efficiently. They set the foundation for the entire manufacturing process and ultimately impact the quality and availability of the final product. Let's talk about harvesting, which is really the bridge between the upstream production and the downstream purification. This step is crucial because it sets the stage for everything that follows. First, we have cell separation. This is where we use techniques like centrifugation or filtration to gently but effectively separate the cells from the culture broth. The goal here is to do this with minimal product loss because every bit of the product is valuable. Next, we move to product collection. Depending on whether our drug is secreted into the broth or remains inside the cells, we'll collect either the broth containing the product or the cell lysate. This transition point is critical because it directly influences the efficiency and the quality of the entire downstream process. In essence, this step determines how smoothly and effectively we can move forward to purify the drug. So paying close attention here makes all the difference in the success of the overall process. Jetzt sprechen wir über den Abschnitt der Downstream-Prozessierung, also die Reinigung und Verfeinerung unseres Products. Zuerst kommt die Clarification, bei der wir Zelltrümmer und unlösliche Partikel entfernen. Das Mädchen wir meist durch Microfiltration oder Zentrifugation, das Material für den nächsten Schritt vorzubereiten. Danach folgt die Capture Chromatography. Hierer konzentrieren wir das Zielmolekül und reinigen es erstmal, oft mit Affinitäts- oder Ionenaustauschmethoden. Das hilft, unser Produkt aus der komplexen Mischung herauszufiltern. Im nächsten Schritt, der Intermediate Purification, entfernen wir weiter Kontaminanten wie Wurzelprotein, Viren oder ihr unerwünscht Stoff. Davor setzen wir mehrere Chromatographies ein, die Reinheit deutlich zu erhöhen. Zum Schluss kommt des Polishing. Hierer erfolgt die letzte Reinigung, alle winzigen Respuren von Warenreinigungen oder Isoformen zu entfernen. Ziel ist es, ein Produkt zu erhalten, das sicher und wirksam ist. All dies Schritt sind entscheidend, sicherzustellen, dass unser Endprodukt den hohen Qualitätsstandards entspricht, die wir ein biopharmaceutisch Produkt stellen. Now, let's look at the final steps involved in preparing the drug product. First, we have ultrafiltration and diafiltration. These processes are crucial for concentrating the purified drug and exchanging buffers, which helps stabilize the molecule for long-term storage. Essentially, we're making sure the drug is at the right concentration and in a buffer that keeps it stable over time. Next comes formulation. This step involves adjusting the pH, adding excipients, and ensuring that the drug remains stable during storage and when administered to patients. Proper formulation is key to maintaining the drug's effectiveness and safety. Finally, we perform sterile filtration. This is the last filtration step before packaging. It's essential for ensuring the final product is sterile, especially since it will be used in clinical settings. All of these steps are carried out in controlled clean room environments to meet strict sterility standards. Together, these steps are fundamental in transforming the purified molecule into a safe, stable, and effective final drug product. Let's now turn our attention to quality control and validation throughout the entire process. This is a critical aspect that ensures the safety and effectiveness of our pharmaceutical products. First, monitoring critical parameters is essential. We need to keep a close eye on factors like purity, potency, yield, and safety at each stage of production. To do this effectively, we rely on advanced analytical methods that provide precise and reliable data. This helps us catch any issues early and maintain high standards consistently. Next, regulatory compliance plays a vital role. We must ensure that all our processes meet the rigorous standards set by agencies like the FDA and EMA. This involves thorough documentation and adherence to established guidelines, which not only supports regulatory approval, but also reinforces our commitment to quality. Finally, process optimization is an ongoing effort. We aim to balance cost, efficiency, and product quality through continuous improvement initiatives. By constantly refining our processes, we can deliver better products more efficiently, ultimately benefiting patients and our organization. All of these elements, monitoring, compliance, and optimization, are interconnected and essential for delivering safe, effective, high-quality pharmaceuticals. They form the backbone of a robust quality assurance system that underpins everything we do. Let's now take a closer look at some of the key challenges and innovations in upstream and downstream processing. Starting with upstream challenges, one of the main focuses is cell line optimization. Improving our cell lines is crucial because it directly impacts the productivity of our bioprocesses. The goal is to develop cells that produce higher yields, which ultimately makes the entire process more efficient and cost-effective. Another challenge is scalability. Moving from lab-scale experiments to full-scale commercial manufacturing isn't straightforward. We need to ensure that processes developed in small batches can be reliably scaled up without losing performance or quality. Contamination control in bioreactor systems is also a critical concern. Contamination can lead to product loss, delays, and increased costs, so maintaining sterile conditions at all stages is essential for consistent and safe production. Switching over to downstream challenges, efficiency in separation techniques is vital, especially when dealing with complex mixtures. The goal is to isolate our product as cleanly and quickly as possible to reduce time and costs. Reducing product loss during purification is another key point. Every step in purification needs to be optimized to minimize waste and maximize yield, ensuring we get the most out of our starting material. Environmental impact and waste reduction are also increasingly important. It's not just about process efficiency, but also making our operations more sustainable and environmentally friendly. Now, on the exciting side, emerging technologies are making a real difference. Single-use bioreactors offer great flexibility. They allow us to adapt quickly to different production scales without the need for extensive cleaning and sterilization, which saves time and resources. Continuous processing is another game changer. Instead of batch processing, continuous methods enable us to produce more consistently and efficiently, reducing cycle times and improving overall productivity. Lastly, advanced chromatography resins are enhancing the selectivity of our separation processes. These innovations help us achieve higher purity levels more efficiently, which is crucial for meeting quality standards. All these challenges and innovations are interconnected and play a vital role in advancing bioprocessing. They help us produce safer, more effective therapies while also addressing the economic and environmental demands of modern biomanufacturing. Let's take a closer look at this case study on monoclonal antibody production. In the upstream phase, we engineer CHO cells, which are Chinese hamster ovary cells, and grow them in large-scale bioreactors. These cells are designed to produce therapeutic antibodies consistently, ensuring high quality from batch to batch. Moving into the downstream phase, we perform a multi-step purification process. This is crucial for removing impurities and achieving a high level of purity that meets stringent quality standards. When we reach the clinical outcome, these biologics are in high demand because they maintain consistent quality and safety profiles. They are used to treat serious conditions like cancer and autoimmune diseases. Overall, this slide highlights how monoclonal antibodies are a rapidly expanding area in pharmaceutical manufacturing, showcasing the importance of integrated upstream and downstream technologies to produce effective, safe therapies. As we come to the final slide, I want to highlight the importance of the symphony between upstream and downstream technologies. Biological transformation is at the heart of this process, where we convert raw biological materials into life-saving pharmaceuticals through precision biotechnology. This step is crucial because it sets the foundation for the entire manufacturing process. Continuously innovating in this space allows us to achieve higher yields, improve purity, and reduce costs. Cutting-edge process development is what drives these improvements, making therapies more accessible and affordable. Now, mastering both upstream and downstream phases is essential for successful drug manufacturing. It's not just about the technical processes, but about ultimately improving patient outcomes worldwide. When these phases work seamlessly together, we can develop and deliver therapies faster and more efficiently. Looking ahead, the future of pharmaceutical manufacturing truly hinges on this integration. By uniting these technologies, we can accelerate development timelines and expand access to breakthrough therapies for more people around the globe. It's about creating a more connected, innovative, and patient-centric approach to medicine.